Welcome to today's chemistry lecture. In this lecture, we are going to study the remaining two factors that affect the rate of reaction, which are effect of catalyst and enzymes as catalysts. So, let's start our today's lecture without wasting any time. So, the fifth effect is effect of catalyst. Let's see the definition of a catalyst: a substance that alters the rate of a chemical reaction without being consumed or permanently changed during the process. How they lower the mechanism on activation energy. A catalyst functions by introducing an alternative pathway for the reaction. This new pathway has a substantially lower activation energy than the uncatalyzed pathway. How it will impact on moles? Because the activation energy barrier is significantly lowered, a much larger fraction of reactant particles possess energy to cross it, rapidly accelerating the product formation. Let's see the graphical representation. Where is an uncatalyzed pathway represents the higher energy curve peak where reactants require a high amount of energy to reach the transit higher energy state. There is another catalyzed pathway. It represents the lower curve peak. The peak of transition state is pulled down, making it easier for reactants to turn into the products. Now let's see how it affects the hydrogenation of ethene. The catalyst we will use is platinum metal. How it works? When adding hydrogen to ethene, both reactant molecules are absorbed onto the physical surface of platinum metal. The results are the chemical bonds within the hydrogen and ethene molecules become weakened on the metal surface. This structural weakening allows them to react together with minimal effort. Now let's see the last factor that affect the rate of chemical reaction, which is enzymes as catalyst. Definition: enzymes are biological catalysts made up of complex protein. Their role: they are responsible for speeding up the highly specific essential chemical reactions taking place continuously inside living organisms. The mechanism: active site. Enzyme possess specialized structural pockets called active site. Reactant molecules known as substrate bind directly into these active sites. Once bound, the targeted chemical bond within the reactant molecules are sufficiently weakened, enabling the reaction to occur swiftly at body temperature. After that, the newly formed product then dissociates from the active site. The enzyme emerges completely unchanged and is immediately free to bind another reactant molecule. Specificity: active sites are highly shape specific. Due to this geometric restriction, a particular enzyme can catalyze only one specific chemical reaction. Here comes our today's quick check problems. First question is, why is a catalyst mostly used in finely divided form? So answer is that just like solid reactants, catalyst works via surface contact. Using a catalyst in a finely divided form like a powder exposes a significantly larger surface area, providing more available sites for reactant molecules to absorb and react simultaneously. Question number 2 is, how do you increase the number of successful clients? So, increasing concentration pressure brings particles closer together, increasing total client frequency. Second, increasing temperature gives particles more kinetic energy, so a higher friction can surpass the activation energy barrier. Adding a catalyst lowers the activation required, allowing more everyday client to be energetic enough to be successful. Third and the last question is, how does the concentration of the products change during a reaction? Explain with the help of a graph. So let's see. At the start of a reaction, time will be equal to 0. The concentration of product is exactly 0. As time moves forward, the product concentration increases rapidly, indicated by a steep upward curve. Over time, as reactants are used up, the rate of product formation slows down, causing the curve to leave off. Eventually, it flattens into a horizontal line when the reaction stops completely and the product concentration reaches its maximum value, as you can see in the picture 17.2. Our today's lecture ends now. Hopefully, you understand all the lecture carefully and remember well.